Thank you everyone for joining us and welcome to the Cancer Research Institute's webinar series, Cancer Immunotherapy and You. Today's date is Wednesday, March 23rd, 2016. The title for today's webinar is Using the Immune System to Treat Colorectal Cancer. In this webinar, we will discuss current immunotherapy treatment options for patients with colorectal cancer. This webinar series is presented with generous support from Genentech and LabAnswer and its employees, along with generous support from Celldex and Regeneron. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. The Cancer Research Institute is the world's only nonprofit organization dedicated exclusively to funding research aimed at harnessing the immune system's power to conquer all types of cancer. We fund research around the world, and the scientists whose work we fund have had discoveries that have led to significant breakthroughs in treating cancer with immunotherapy. Over the next 30 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how these breakthroughs are transforming the treatment of colorectal cancer. After a 15-minute presentation from our speaker, we'll open the discussion to questions submitted by you. You can pose your questions at any time by typing in the Q&A box on your screen. You can also submit questions online by tweeting them to us at Cancer Research using the hashtag CRIWebinar. Today's webinar is being recorded and will be made available on the CRI website and on our YouTube channel. Now it's my pleasure to welcome today's expert speaker. Dr. Neil Segal is a medical oncologist who specializes in the treatment of patients with gastrointestinal cancers. His research interests focus on the development of new therapies. In particular, he is investigating innovative ways to use the immune system to treat cancer. Dr. Segal, we are grateful that you could take time out of your day to join us, and welcome.
Brian, thank you very much for that introduction, and it is truly my pleasure and my privilege to be talking to you all today in affiliation with the Cancer Research Institute, who have been pivotal in advancing immune therapy research and is a very important institution toward furthering immunotherapy for cancer. So it's really my pleasure to be here. Today I'm going to talk about colorectal cancer, which is very important to me and is a significant challenge that we're facing as we strive toward creating better therapies for our patients, to enable patients to respond to immune therapy and to create new treatments to help our patients. And in terms of the overview, today I'm going to discuss immunotherapy for colorectal cancer. I'm going to focus on what's called MSI-high colorectal cancer. I'm going to look at why immunotherapy can work for colorectal cancer, and in particular MSI-high tumors, and I'll explain in a few slides exactly which patients that includes. And we'll look at some results of clinical trials, and we'll look at some of my newer research which gives some insight into where the future of colorectal cancer immunotherapy is really heading. So I like to start off any discussion about immunotherapy to look back on the last many years, more than 100 years, from 1893 up until 2016, which reminds us how much we have moved ahead in immunotherapy, but it also reminds us how much work we still have to do. So just looking back, it's many years since 1893 when William Coley first found that if a patient has inflammation around the site of a tumor, in some cases that tumor could regress. That was really anecdotal observations of how this worked and led to bacterial toxins. And really the essence of what we knew back in the 1800s was that if there's inflammation, tumors can respond. That was the first insight that the body itself can attack cancer. And we've really learned how this works, and now we can make it better, and we can make it more focused. So certainly there's not bacterial toxins being injected, but we understand the mechanisms, and that's where we focus further treatments. And then about 23 years later, in 1909, or 16 years later, excuse my calculations, Paul Ehrlich proposed something called the immune surveillance hypothesis, which enabled us to understand that the immune system essentially can move around the body. It can identify where cancer is, and in many cases it can attack the cancer, and in other cases it needs us to help it make it work better, which is what immune therapy is. And then in the 1970s and 1989 we identified that there's a coordinating cell called the dendritic cell, and afterwards in the 1890s we identified these key checkpoints, which is occur between the dendritic cell and the T cell. And the analogy that I like to use is that we think of the immune system really as an army, and there's warriors and there are soldiers, which are the T cells, the immune cells, which will actually go out and attack the cancer. And then there are these generals, which are the dendritic cells that coordinate the immune response. And there's a lot of interplay between the dendritic cells and the T cells, and as well as other types of immune cells, which results in a war. And this is the immune system which is attacking the cancer, and we can manipulate this and make it better, which is where therapy comes in. And we really saw that in the 1990s with Jim Allison showed in the lab that by blocking an antibody, that using a blocking antibody against an inhibitory checkpoint, which is an off switch, CTLA-4, can fight cancer. And this led toward one of the first clinical trials showing that this works in melanoma, and since then there has been many opportunities to develop further immune therapy. One of the pivotal questions is, why immune therapy? We talk about chemotherapy, we talk about radiation therapy, we talk about many different treatments, but why are we talking about immune therapy? And there are three important points. The first point is that the immune system is universal. It is capable of recognizing a near unlimited array of markers and attacking them. When we set these soldiers, these white blood cells, out to attack something, they look for a marker, right? They want to see what they should attack. Initially, they're trained to fight bacteria and viruses, but they can also fight cancer. So they look for markers. And there's many different types of markers. So we could use immune therapy for every type of cancer, or we can do research to learn how it works. The second point is that the immune system is very specific. It has this ability to distinguish self from non-self. The way that it does this is by identifying what these markers are. And we'll talk a little bit later about how the immune system is able to distinguish self from non-self, because ultimately what we want is the immune system to attack the tumor, but we don't want the immune system to attack normal tissue, because that would be a side effect, and that would be inflammation, and those are the types of side effects that immune therapy can result in, but we want more of a specificity against the tumor. And the third point is that the immune system has memory, which means it can rapidly respond to an antigen or a marker that it previously encountered. So even if the immune system had seen a marker like that was found on a cancer, we can retrain it to rapidly go back and attack the tumor. So these are three salient features of the immune system which have enabled us to develop immune therapy. And we're going to take a little step to talk about mutations and what is a mutation, why is it important, and we'll digress to discuss hereditary cancers and how this plays in. Now, what you see here in front of you is a diagram that illustrates what a mutation is and enables us as researchers and physicians and patients and people that are interested in immune therapy to understand why a mutation is a marker. So you can think of DNA as being the blueprint that's present in every cell, and that this is an example here. Let me get the arrow. And here you see a DNA sequence, and there are long strings of these sequences. But what this specific sequence and the order of these letters does is it codes for a protein. So the cell will make proteins based on this DNA sequence, and the types of proteins that are present in the cell will give the cell its characteristics. Should it be a muscle cell? Should it be a skin cell? That's really dependent on the types of proteins, and there are some proteins that control how cells divide, and those are generally the types of mutations that unfortunately will lead to a cancer. Now, if this is the normal DNA sequence, in cancer there is a mutation, and the mutation is a feature that happens in cancers, and there are many mutations in cancer. But what happens here is that when this makes a protein, the fact that there's a change, that this T is now something different, then the immune system will recognize that protein as being something that's new, that developed during the cancer. It shouldn't be there. The immune system wants to attack it and wants to destroy it. And what we did was just a study where a proof of principle. We took approximately 12,000 genes. We took the DNA sequence from 12,000 genes, in 11 different colon cancers, and we found that on average there's about 7 novel markers due to DNA mutation, and these novel markers can be recognized by the immune system. And that was a theoretical study, but it was really a proof of principle. And we realized that we needed to study immunotherapy in colon tumors that have many mutations specifically. So on the next slide here, this is an illustration of mutations in cancer. And this is here we have about 20 different tumors. And this was a study done by a group in London, in the UK. And each dot here represents a tumor, but the dot represents the number of mutations on a log scale. So there's 10 times more mutations as you go up each graph here. And this is the distribution of how many mutations occur on average in each tumor. And if this graph, if the red line is low, it means there's fewer mutations. If the red line is high, it means there are many mutations. And an interesting observation here is that the tumors with many mutations, melanoma, lung cancer, bladder cancer, are the types of mutations that can respond to immune therapy. And this reminds us that the cancers with more markers can be attacked by the immune system. And that's a correlation. But what we also see is that colon cancer is very close, and it's even closer than other kinds of cancers where there's been some reports so far of some responses, stomach cancer, head and neck cancer. So there really is a potential here for the immune response to occur against colon cancer. But within colon cancer, there's a type that have many, many mutations, and these are the MSI high tumors. And we'll get to those in just a minute, and we'll talk about that. So we've spoken so far about the markers that the immune system is going to attack, and now we're going to digress slightly to talk about the different types of immune therapy. And the first type of immune therapy is called cytokine therapy. And cytokines are proteins that are made by cancers and also by the immune system, and this is how the immune system talks to itself. And it's almost like a war cry. If there's more cytokines, the immune system is going to be stronger. If it's a cytokine that is really a positive cytokine, there can also be negative cytokines to switch off the immune system. But it's the way that general talks to the soldiers or the soldiers talk to each other. And in some treatments, we can give cytokines to non-specifically activate the immune system. A second type of immune therapy is a therapeutic vaccine. So here we use the vaccine to show the immune system a marker, and that marker could be a mutation, the marker could be a protein that's not mutated, or something that the immune system could recognize, and that might trigger an immune response to go back and identify what their target is. And if this was a flu virus, it would be looking for the flu, but if we use a protein from a cancer, then it will look for that cancer. The next type of immune therapy is adoptive cell therapy, and these can be T cells stimulated in vitro or the modified T cells called chimeric antigen receptor, CAR T cells. And here, doctors will remove the white blood cells or the soldiers from a patient and either grow them here, they'll grow them, or they'll genetically change them and then put them back in the patient with the ability to go in and then seek out and destroy the tumor. The next type is an oncolytic virus. These are viruses that are specially made in a lab, but they can go into the tumor, they can destroy the tumor, and they can also switch on the immune system. And then finally, immune checkpoint blockade. And immune checkpoint blockade is going to be the focus of what we're going to talk about for the remainder of this presentation. And the analogy that clearly illustrates what this is if you think of your immune system as a car, and this car is either going to accelerate or it's going to brake. And these are molecules or these are proteins in the car or on the cancer that will make the immune system stronger, or they'll make it slow down. And an example of the ones that make it slow down are CTLA-4 and PD-1. So if we use a drug which is called a CTLA-4 or PD-1 checkpoint blockade, it removes the brakes, which means as that car gains speed and it becomes stronger, it's going to move in and attack the cancer. So the white blood cell, the immune cell, is what this car is. Now, what has been done so far? What therapies that target CTLA-4 or PD-1 have been used in colon cancer? And the first one that was shown is this drug called Balamap, which is an anti-PD-1. So it takes away the brakes of the immune system, allows the immune system to be strong and go into the cancer. There were two phase 1 trials. A phase 1 trial is a trial intended to determine what the dose is and the side effects of a therapy. And in the phase 1 trial, there were about 20 patients with colon cancer. We unfortunately learned that in 19 of these patients, the cancer did not shrink. And this was the first insight that nivolumab anti-PD-1 by itself does not work in colon cancer. It works in other kinds of cancers, for example, in melanoma, lung cancer, but not in colon cancer. But what was observed here is there was one patient who had a complete response to this therapy, and they had a microcell unstable tumor. So there was something about this kind of cancer that allowed that patient to respond if they got the anti-PD-1. And this really was the first trial that showed a response in MSI-high tumor as compared to microcell stable, which is really conventional colon cancer. So MSI-high is rare. It's about 4% of metastatic colon cancer. Microcell stable is the remainder. So what this microcell instability is it's caused by loss of a DNA mismatch repair activity. That's the long name for it. And it's much more common in early stage cancer, in stage 1 to 3, where it's about 50% of colon cancers. But as mentioned, it's much less common in cancers that are metastatic and spread. And this was a clinical trial that was done by Young Lee and colleagues. And what they did here is they took patients with microcell instability, colon cancer here. They also had conventional colon cancer, which is called mismatch repair proficient, and then they had non-colorectal cancer patients. And these patients were treated with pembrolizumab, which is an anti-PD-1 drug, which makes the immune system stronger. And here they showed a staggering 62% response rate in 13 patients that with single immune therapy, there was a profound shrinkage of microcellular unstable colon cancers. Unfortunately, there was no confirmed response rate, which is so a response rate is a measure of tumors shrinking or going away completely. So there were no tumors that had substantial shrinkage or that went away in conventional colon cancer, similar to what we saw with the prior trial of nivolumab. But we did see that this really works very effectively in patients with microcellular unstable colon cancers. And this was a very important turning point in developing new therapies and understanding how they work and providing these two patients that have this mismatch repair deficiency. So I'm going to turn now for the next few minutes to some of the trials that are being done at Memorial and in our research program here. So within my clinical research, one of the trials here is this drug Balamap, which is an anti-PD-L1. Right, this is the brake for the immune system. We take away the brakes. We're really focusing on certain kinds of cancers with immunological subsets with certain criteria. And the rationale here is that by taking away the brake, it will increase the immune system's ability to go in and destroy the cancer cells. But here, we're specifically assessing Balamap in patients with Lynch syndrome. Lynch syndrome is a rare syndrome. It's an hereditary syndrome where patients have mismatch repair deficiency, and the tumors have lots of markers and they can respond, as we saw in the previous study. We also are going to include patients who have tumors that look like they have Lynch syndrome, but they don't actually have the Lynch syndrome. They acquired this genetic mutation, but they have lots of markers for the immune system. And then we're going to look at patients with conventional colon cancer, but those that have many white blood cells in the tumor. And that's a rarer type of tumor. But unlike most colon cancers, there really are a lot of soldiers, white blood cells in the tumor. So the question that we're asking is, if they can go there already, can we make the system stronger to try and destroy these tumors? And this is an ongoing study, and if you want more information about this clinical trial, it's on clinicaltrials.gov, and that's the reference number. The second clinical trial that we are doing is a combination of radiation or ablation plus pembrolizumab, which is anti-PD-L1. And the rationale here is that radiation therapy or ablation. Now, ablation is a technique where we use freezing or energy to kill the cancer cells. That this is a standard therapy for the treatment of colon cancer, and it's known from mouse studies and some reports in patients that this can trigger an immune response. And the laboratory studies certainly support that by adding an anti-PD-1 to the radiation therapy or the ablation can increase the immune system's ability to control or destroy the cancer. And so here we're really encouraging patients who are going to undergo radiation or ablation as a standard treatment, for one tumor, and if there's another tumor that's not going to be ablated, then we'll watch and we'll see if that tumor can shrink by giving the pembrolizumab to make the immune system strong after they get the radiation or ablation. And the third trial that I'm going to talk about is this R068958882, which is combined with the atezolizumab, which is an anti-PD1. So that atezolizumab will take off the brake, and the R068 is going to make the immune system stronger. And the way it does this, the R0689582 is an antibody. Antibodies usually recognize as target, and they may by the immune system, but in this case we're making these antibodies in a lab. This is a drug. This is a large multi-center clinical trial that's been done in many sites, and here Roche made this, and this protein, one end of it attaches to a protein on the cancer cells called CA, and colon cancer very often makes this CA, and the other end attaches to a protein which is designed to stimulate the immune system to fight the cancer cells. So here we make the immune system stronger, and we now give it this atezolizumab, the second point, which improves the immune system's ability to recognize and destroy the cancer cells by taking away the brake or blocking PD1, which is this protein made by cancer cells. And what we're hoping is that these two drugs together will trigger an even stronger immune response against the cancer. And if you would like more information about that clinical trial, you can go to clinicaltrials.gov, and that is the reference number. So in summary, at least two immune subtypes of colorectal cancer exist. There's this MSI high colon cancer, which has many markers, and immune responses are really prevalent, and we can make these stronger with an anti-PD1 or potentially anti-PD-L1, and they can be benefit. And then there's this microcell stable conventional colon cancer, which for reasons of biology, which we don't really understand yet, are immune resistant despite having a similar mutation burden to other immune responsive cancers, which I showed earlier on, that they have as many mutations as bladder cancer, head and neck cancer, yet they don't respond as well. So what we need to do going forward for immunotherapy to become effective in the treatment of colon cancer, we are going to continue exploring studies in microcell instability high colon cancer, the MSI high colon cancer, to make the responses even better. And we also really need to focus on alternative strategies, and these are being explored in conventional colorectal cancer, including things such as radiation or ablation to make the tumor more visible to T cells, and then we release the PD-1 brake, or we would use cytokines, these communicators with the soldiers with the immune system, to harness the T cells, to bring them to the tumor, to switch them on, and then combine it with an anti-PD-1 so that the tumor doesn't suppress them. But in general, other novel targets and emerging therapies are being studied, and this is the way forward from this point going onwards. So really, thank you for your attention and for listening to me for the last 20 minutes or so.
Well, thanks very much, Dr. Segal. Excellent presentation so far. And I'd just like to remind all those of you who are watching right now that you can submit questions to Dr. Segal by typing in the Q&A pod. We're limited in time, so we'll unfortunately not be able to get to all of the questions that you submit, but I strongly encourage you to do that. And so we'll start with our first question from someone who's viewing. Dr. Segal, so you're talking about the mutations, and that higher levels of mutations in some colorectal cancer patients means that they will be more likely to respond to immunotherapy. How does a patient learn whether or not their colorectal cancer has those mutations? And is there a test that's done? Is this routine, or does a patient have to ask for this test to be done?
So the test that is done is called the MSI high, which is a DNA analysis test, or there's a stain that can be done in the tissue. If there was a biopsy done and there's tissue in the path lab, that can be obtained, and that can be tested for changes in some proteins. Either the DNA test or the tissue test can indicate that the tumor has this can be MSI high with many mutations. This is not necessarily a routine test. This test is usually done if there's a suspicion for the hereditary syndrome. So in people with a certain family history where there's lots of cancers or other colon cancer or certain other kinds of cancers that can be part of this syndrome, or if they're younger, or if the pathologist is concerned that the tumor might look like it has this syndrome. So there's usually a trigger to do the test. So it's not always done. And I would really encourage patients who have questions about doing this MSI-High test to discuss with their oncologist whether the test is indicated for them and could be helpful. So we don't do it for the purpose of immunotherapy, we do it for the purpose of usual standard of care.
I see. You mentioned toward the end, in terms of future steps, that there's still a great amount of work that needs to be done, not only to improve the responses in patients who do have high levels of these mutations, but also to make colorectal cancers more immunogenic or capable of being seen by the immune system in patients who have low levels of mutations, and you discussed radiation as a technique for that. Can you say just a little bit more about where that research is going and what's the rationale for it?
So the area that the research is going is by looking at novel therapies, and there's many novel therapies that we're learning about and that are being developed that can make the immune system stronger. So we want to apply these novel immune therapies in patients with colon cancer, either by themselves or combined with an anti-PD1 or anti-PD-L1, which takes away the brakes. So the strategy is to educate the immune system, make it stronger, and potentially at the same time to remove the brake to allow the immune system to go in. We may not always need a combination, we may need one or the other. So trials might have an arm where there's one drug, and they might have an arm where there's a combination therapy. The benefit of a combination therapy is it may work better. The downside is that there may be more side effects. So it's very important to study both of those. Radiation therapy is just one example of a strategy to ignite the immune response, but there are other ways to do it.
Thank you. You mentioned CAR T-cell therapy in your presentation, and these types of therapies have been successful in certain blood cancers, but much less successful in solid tumors. So can you tell us a little bit more about what's happening in CAR T-cell research in colorectal cancer?
So CAR T-cells means that the immune system, the white blood cell, has been modified so that it can directly get into the tumor, and that's been done by changing the receptor. And we can make the receptor specific. The receptor is the way the white blood cell sees the marker, and we can make the white blood cell see a marker that's found on a leukemia cell, for example, and that's where CAR T cells can attack leukemias. In solid tumors, it's been more challenging. The results have not been as promising, and we don't really understand why just yet, and that may be beyond the scope of this. But in colon cancer, one of the markers that's been explored for CAR T cells is the CEA. I spoke a little bit earlier about CEA as being the anchor for this the cytokine, and CAR T cells against CEA have been tried. They're in just a handful of patients. Some of the data looked encouraging, but there were lots of side effects. So CAR T cells may have a place in the future therapy for colorectal cancer, and I certainly encourage them being studied. But we need to learn from our colleagues who are developing CAR T cells in other kinds of cancers how best to make these work before we know their future in colon cancer.
At what point in a patient's treatment of colorectal cancer does he or she either become eligible to be treated with immunotherapy? When does immunotherapy enter the formula for patients?
If it's an immunotherapy in an early phase clinical trial, then in general, patients would have been treated with at least one standard therapy for cancer that has spread. So clinical trials usually begin in tumors that have spread, and there's several reasons. One of the important reasons of doing an immunotherapy trial in a cancer that's spread is that we can do a scan and we can follow that tumor and we can see whether or not the immunotherapy is working. So it gives us an indicator of how well the therapies work. And there's eligibility criteria for clinical trials which are designed to make sure that it's really safe and appropriate for the patient to be on the trial. And in general, we want a patient to have had at least one standard therapy for their metastatic cancer, whether it be FOLFOX or FOLFIRI. Some clinical trials will require more than one. Those are the usual types of therapies. I think it's an important conversation that all patients should have with their oncologists at the time that they're starting therapy, is at what point should they be looking for clinical trials, including immunotherapy trials.
For patients that are treated in community oncology offices rather than intense research institutes, will those oncologists also, like, has immunotherapy entered the standard of clinical practice in community oncologist offices?
So that's a good question. Immunotherapy is a standard treatment for many kinds of cancers, including melanoma, for lung cancer. It's effective in renal cancer. There are therapies, immunotherapies that work in these kinds of cancers. So community oncologists already have a very good understanding and ability to administer immune therapy. Some of the newer immune therapies, so the ones that are approved so far take away the break. Some of the newer immune therapies, which are making the immune system stronger, are newer. The community oncologists may not be using them as a standard therapy, but certainly these trials could be available. It depends whether that community oncology setting is running clinical trials. If they are, then these trials will eventually become available. If not, then they will be available at more academic centers.
Okay. Another type of mutation is the KRAS mutation. Wondered if you could say anything about what kind of progress is being made in treating patients with a KRAS mutation. And also something called IDO inhibitor immunotherapy, if you could say a little bit about that.
So a KRAS mutation is one of the many mutations that occurs in some colon cancers. So beyond KRAS, there's a whole extended RAS profile of similar mutations. And what we know is that if there's a mutation in the cell, it means that certain standard therapies, cetuximab, panitumumab, won't work if there's a mutation, because the cancer will continue to grow. And the mutation is inside the cell. When immune therapy is designed to attack the cancer, it should work independent of whether or not there's a RAS mutation. So the majority of clinical trials that are being done now in immunotherapy for colon cancer are not pre-selecting patients based on whether or not there's a RAS mutation. So regardless. However, patients that don't have a RAS mutation would have more standard therapies than patients with the RAS mutation. So they're both, but at some point, they're both good targets for immune therapy trials. There's not data yet whether the RAS mutation will really matter for immune therapy, but I don't think it will. IDO is one of the newer types of immune targets, and an IDO inhibitor could help with an immune response. And it's too early now to know what the role of an IDO inhibitor is going to be, but I would consider this as one of those armamentarium of future immune therapies trials for colon cancer.
Great. This is an interesting question. Does age factor into whether or not a patient is likely to respond to immunotherapy?
That's a good question. At some point, we'll know whether age is a factor. I don't think that age is a factor now for response to immunotherapy. In tumors that do respond, not colon cancer, we've seen responses in older patients that have had very good responses. In fact, when I give a talk about immunotherapy, I often show a figure of a lady in her 80s and above who was on a clinical trial who had a profound response. So it's not, what's more important than age is just generally how well the patient will be able to get through the treatments, and which we call something called the performance status. So immunotherapy has a role for all patients regardless of age.
Well, that's all the time that we have today, Dr. Segal, and those of you watching. I want to thank all of you again for participating, especially you, Dr. Segal. You mentioned clinical trials and the importance of clinical trials, especially in colorectal cancer research. And in addition to the clinicaltrials.gov database, which you highlighted, I'd also like to point out to the patients and caregivers that are watching right now that we have a website that's devoted entirely to you, and it features information on cancer immunotherapy for various types of cancer. Also includes a clinical trial finder service So it's a phone number that you can call and speak to a navigator who will help try to connect you with an immunotherapy clinical trial for your type of cancer. Also, it features stories of patients that have been treated with immunotherapy. So if you'd like to learn about, from the patient perspective, what the experience is like, this is a resource to you, and you can go to it at www.theanswertocancer.org. Once again, I'd like to thank our presenting sponsors, Genentech and LabAnswer and its employees, and our generous support received from Celldex and Regeneron. Thank you very much for that. I'd also like to thank the folks at Fight Colorectal Cancer, which is a patient advocacy organization devoted to the colorectal cancer patient community. They've been very helpful in spreading the word about today's webinar, and so we appreciate partnering with them as well. Thank you all, and if you will, please, we'd love to get your feedback just after this. You'll be directed to a very brief survey. Those surveys are extremely important to us to make sure that we're providing the kind of content that is valuable to you. So with that, thank you all very much. Thank you, Dr. Segal, and be well, everyone.
Thank you.